Hi, class. Okay, so protein synthesis in detail. We're going to talk about a more detailed look about how proteins are made. In the last video, we kind of introduced it. We talked about the initial steps of DNA to RNA is transcription, RNA to a protein is translation, but now we're going to get into actually exactly what's going on behind the scenes with protein synthesis. Okay, so like we mentioned before, it all starts with DNA, right? DNA has got the code for how to make certain amino acids, and you don't really know what this means yet, but remember that this is a code, and just like any other code, it may not make sense unless you have some way of decoding it, which is what we're going to learn how to do today. So as we know, DNA is two backbones of nucleotides. Every nucleotide has, every nucleotide has a phosphate group, a sugar molecule, and a third thing that's the base. Now that base could be either a G, a C, an A, or a T. So this is phosphate sugar G, phosphate sugar C, phosphate sugar A, phosphate sugar T. And across from it, as we know the base pairing rules, car in the garage, C and G always go together. Apple in the tree, A and T always go together. So this code here, we're going to learn how to decode this today. So let me actually just finish this out here. I'm going to make 6 letters long. That'll make sense here in a minute why I'm going to do that. But remember from the last video, step 1 is called transcription. So in transcription, I'm running out of room already. In transcription, that's when the code that's in DNA gets rewritten into RNA. And last time I just told you that it goes from, DNA to RNA. I didn't specify. Well, today I'm going to specify which kind of RNA this is. And the kind of RNA that this DNA is going to be rewritten or transcribed as is mRNA, right? There's different kinds of RNA. mRNA is one. There's tRNA. And then there's rRNA. Okay. Now we're not going to talk about rRNA. We're just going to talk about M and T. So when I said last time that DNA gets rewritten into mRNA, it's, I said it just gets rewritten into RNA. The specific kind of RNA it gets rewritten into is mRNA. And the M stands for messenger. Okay, so this is messenger RNA because it's going to carry the message from DNA to the place it needs to go. So first of all, why do we need messenger RNA? If you remember from last time, DNA is stuck inside the nucleus of the cell. So remember, every cell has a nucleus, and DNA is trapped in here. He's trapped in here and he cannot leave. The problem is that the place where the proteins are actually made is outside of the nucleus in these places that we call ribosomes. Okay, so remember, DNA has the instructions and the information on which proteins to make and how to make them, but the place where they're actually made is outside the nucleus where the DNA cannot go because he's trapped in there. So how does DNA get the message from inside the nucleus to the ribosomes outside the nucleus? Well, that's where mRNA or messenger RNA comes from, okay, or comes in. So a strand of messenger RNA is going to be created inside the nucleus, and then that messenger RNA, let's say this is messenger RNA, a little squiggly, is going to copy the message from DNA, deliver it to the ribosomes, and then that's where the protein is actually going to be put together. Okay. I'm making sure I stay on track here. And once again, that's called transcription. So transcription is where this is what this is called, and it happens inside the nucleus. Okay, that's rewriting DNA into a strand of messenger RNA. Now that messenger RNA is then going to deliver it to the ribosomes and get created to create the protein, and that's what we call translation. So translation is when mRNA delivers the message to the ribosomes and the amino acids get put together. Okay, so the ribosome is the place where this happens, which is these things here, right? Okay, that's where the amino acids get put together to make the actual protein. And then we end up with a protein. But this still doesn't explain exactly what's going on. This is just kind of a brief overview of the overall process. So now let's get into the nitty-gritty of exactly what's happening. So I'm going to erase this. Okay. So once again, we start off with a double-stranded DNA molecule. And on one strand, we have nucleotide bases, phosphate sugar A, phosphate sugar T, so on and so forth. And on the other strand, we have nucleotide bases. And according to the base pairing rules, this is what will be on the other strand. Okay, so let's call this strand number 1 and strand number 2. So what happens is that this DNA strand, one of these two DNA strands is going to get read and rewritten into a strand of RNA. Remember, RNA is only single-stranded. So we're going to copy one of the two DNA strands. We're going to copy the instructions in that strand and then basically print out a new mRNA strand. But there's an enzyme that does this, okay? So remember, enzymes are one of the many kinds of proteins. There's an enzyme that helps make this happen. And that enzyme is called RNA Polymerase. Okay. So let's say the enzyme, this is the enzyme RNA polymerase. So the job of this enzyme, RNA polymerase, is to go right between the two strands of DNA, which, by the way, these two strands are actually loosely held together by these weak bonds that we haven't talked about yet, but that I'm going to tell you about right now, which are called hydrogen bonds. Okay, so these hydrogen bonds are kind of like these weak little bonds that kind of keep things together, but you can easily pull them apart, put them back together again. So RNA polymerase is going to go between the two strands, separate the two nucleotides by breaking these hydrogen bonds that are keeping them together. And as it does that, it's going to read each nucleotide on the DNA strand and print off an RNA complement. First of all, the strand that gets read, the strand of DNA that gets read is called the template strand. And let's say strand number 2 is the template strand. That's the one that's going to get read and copied. Now, copy is not really a good word because it's not an exact copy. Whenever we copy the instructions, when RNA polymerase copies the instructions from DNA and prints out the new mRNA strand, it's not really making a copy. What it's doing is it's adding the complement to each letter or each nucleotide. So I'm going to make the RNA strand red. RNA polymerase is going to read a T, but it's not going to just copy the T. It's going to print out what goes with the T, which is the tree, right? What goes with T is A, apple in the tree. Okay. We're going to print off not the A. We're not going to copy the A. We're going to print off what goes with the A, which you might think is a T because A and T, apple in the tree. But remember that DNA is GCat, but RNA is GCal. Remember, there's a U instead of a T. So everywhere we would otherwise put a T if we were working with DNA, you have to remember I'm not working with DNA, I'm working with RNA. So everywhere I would normally put a T, I'm going to put a U instead. So normally we would put a T if it was DNA, but because it's RNA, we're going to put a U instead. C, we're going to read the C, and we're going to print out the complement to the C, which is G. Remember, RNA still has a G. Okay, the only letter you change is T into U. That's the only one. Everything else is still the same. T gets rewritten as an A, G gets rewritten as a C, and A gets rewritten as a U. Okay, so this is now our new strand of messenger RNA that we created using the enzyme RNA polymerase reading the template strand on the DNA. Okay, so now we have our mRNA. Now the mRNA has to leave the nucleus and go to the ribosome. So we're in the ribosome, which by the way, kind of looks like a weird little, it kind of looks like a hamburger bun, like it's got a big part on top and a small part on the bottom. Okay, so this is the ribosome. It's got two parts, the top bun and the bottom bun. The top bun is bigger and the bottom bun is smaller. That's not necessarily important for you to remember. I'm just telling you why I drew it this way. So now what we have to do is we have to break this up the way that we decode this, and we're ready to decode this now. This is where the decoding comes in, okay? So the decoding, the first step in decoding this is to break up the mRNA strand into chunks of 3. So here's a chunk of 3, and here's a chunk of 3. And each chunk of 3 is what we call a codon. So this is codon number 1, and this is codon number 2. You'll notice it's called a codon because we're decoding. This is a code, right? So that's where the code comes from. It's a codon. So codon 1, codon 2. And the thing about codons is that every codon is going to tell you to make a specific amino acid, which we're going to do right now. So now we're ready to use our decoder chart. Now that we have our RNA strand broken up into codons, now we're ready to decode. And that's where this thing comes in. Let me introduce to you the fancy schmancy, let me change the red first, fancy schmancy codon chart, okay? So let me go back and remind myself what the codon was. So we got AUG, okay, so AUG is the codon, all right? AUG, so this is the decoder chart. On this chart is a list of the 20 different amino acids that make up all proteins. We previously learned that there are 20 different amino acids. All proteins are chains of amino acids, like a beaded necklace. Well, these are the 20 kinds of beads, okay? These are the beads that you can put on the necklace to create the necklace, right? So the way that you find out which bead or amino acid to put on first is you look at the codon, AUG, and you find the first base first on the left-hand side, which is A. So the first base is A, right? So we find A, and what that does is it tells you that it's in this row. So we know that it's going to be somewhere in this row. Then you go to the second base. The second base is U. You look up the second base on the top. Okay. Find the U. That tells you it's in this column. So we now know that it's in this row and this column. So it's somewhere in this box right here. But which of these four in the box is it? Well, that's where the third base comes in, G. So we find the third base, G, and we follow it, and here we are, it's methionine, okay? So AUG tells you to make methionine. I'm just going to put MET for short, okay? So hop back over here. The first amino acid in our sequence of amino acids, which all put together make up the protein, is methionine. And now we have to do the same thing with the second one. So we've got ACU. So let's hop back over. Let's find out what ACU makes. Let's start with A. Let me do this in blue just because that way it's easier to tell the difference. Okay, so A, so we know it's going to be in that row, right? C, we know it's going to be in this column here. So we know it's somewhere in this box. And not that it matters because all four of the amino acids listed in this box are all 309. But just so we can say that we did, we'll look up the last letter, which is U, and follow that. And it tells us it's in that specific spot there. So we now know that ACU makes 309. So I'm going to put THR for short, okay? And we're going to, now if we had 1,000 codons, we'd have 1,000 amino acids, but I'm obviously not going to do that, okay? So I talked to you earlier about mRNA, but this is where tRNA comes in, okay? So I mentioned that I was going to talk about tRNA as well. So tRNA, let's make tRNA blue. Why not? I've already got blue up here. So tRNA is what we call, excuse me, transfer RNA. The T stands for transfer. I'm losing my voice. And what transfer RNA does is transfer RNA is the thing that actually delivers the amino acids. This is the delivery boy. This is the messenger boy. This is the delivery boy, okay? The delivery boy delivers the amino acids, these amino acids, to the ribosome where they get put together. Okay, so the messenger tells the ribosome, it gets read by the ribosome. So imagine this is just like a printed off strand of paper. You know, the ribosome is going to read it, but tRNA is the thing that actually physically brings the amino acids. These amino acids are just kind of floating out there in the cell, and tRNA goes and gets these amino acids. So it'll be like, okay, messenger RNA says we need a methionine. We're going to, transfer RNA goes and fetches the methionine amino acid and brings it back to the ribosome. And then the ribosome links that with the next amino acid, which another tRNA is going to come in and bring. It's going to go grab the 309 and then take it back to the ribosome. And then the ribosome is going to put the methionine together with the 309. And then every other amino acid after that. And like I said, all the rest of the 1,000 amino acids, it's going to link them all together. And then whenever it comes out, it gets printed out of the ribosome, and then you have your protein, which is the chain of amino acids, right? So one other thing I forgot to mention earlier, RNA polymerase, anything that ends in ACE is going to be an enzyme for the most part. There's nothing I can think of that ends in ACE that's not an enzyme. So that is the process in detail. Remember, DNA gets rewritten into messenger RNA, that's transcription. The messenger RNA is then translated by the ribosome. They take codons, you break them up into chunks of 3 called codons. Each codon tells you to make an amino acid. The ribosome physically builds the protein by linking those amino acids together. But who brings the amino acids to the ribosome so the ribosome can take them and put them together? tRNA. That's where that guy comes in. All right, confusing enough? Great. Watch more videos. Maybe it'll help.